Welcome to Module 1 of Blood Transfusion Science. In this session, we will begin with the ABO and rhesus blood group systems, which form the very foundation of safe blood transfusion practices. We will then explore how these blood groups are inherited, how they are distributed in different populations, and how we apply genetic theory to understand and predict blood group phenotypes. Next, we will walk through the techniques for blood grouping, including their principles, advantages, and disadvantages. We will dive into the preparation and standardization of antisera, and learn about plant lectins and how they are used in blood grouping. Finally, we will discuss anticoagulants used in blood collection, blood containers, and methods for donor screening, ensuring you understand both the science and the practical application in a clinical laboratory setting. Section 1, ABO and rhesus blood groups. 1.1 The ABO blood group system. The ABO system was discovered by Karl Landsteiner in 1901, and it remains the most important blood group system in transfusion medicine. The classification is based on the presence or absence of two antigens, antigen A and antigen B, on the surface of red blood cells. The four main ABO blood groups are: Group A has antigen A on red cells and antibody B in plasma. Group B has antigen B on red cells and antibody A in plasma. Group AB has both antigen A and antigen B on red cells, no anti-A or anti-B antibodies in plasma. Group O has neither antigen A nor antigen B on red cells, has both anti-A and anti-B antibodies in plasma. 1.2 Rhesus, the RH blood group system. The RH system, discovered in 1940 by Landsteiner and Wiener, is second in clinical importance after ABO. The key antigen here is the D antigen. If present, the individual is Rh positive; if absent, they are Rh negative. Rh positive, D antigen is present on the red cell membrane. Rh negative, D antigen is absent. The Rh system also includes other antigens such as C, E, and E, but D is the most immunogenic and significant in transfusion medicine. 1.3 Inheritance patterns. Blood groups follow Mendelian inheritance principles. ABO inheritance. The A and B alleles are codominant. The O allele is recessive. Genotypes and phenotypes. A or AO blood group A. BB or BO blood group B. AB blood group AB. OO blood group O. RH inheritance. The D antigen is inherited in a dominant manner. An individual needs only one copy of the D gene, from either parent, to be Rh positive. Rh negative phenotype results when both copies are AD, no D antigen production. 1.4 Population distribution. ABO and Rh frequencies vary by ethnic group and geographical location. In many African populations, O positive is the most common. In European populations, higher percentages of A and Rh negative. This information is crucial for blood bank inventory planning. Section 2, Blood grouping techniques. 2.1 Principles of blood grouping. Blood grouping is performed by mixing patients' red cells with known antisera and looking for agglutination. Forward typing detects antigens on red cells using known antibodies. Reverse typing detects antibodies in plasma using known red cells. 2.2 Common blood grouping techniques. Slide method. Quick and simple. Not very sensitive. Useful in emergencies. Tube method. More sensitive and reliable. Requires centrifugation. Gold standard in many labs. Microplate method. High throughput for mass screening. Uses small quantities of reagents. Gel card method. Very accurate. Requires specialized equipment. 2.3 Advantages and disadvantages. Advantages. Early detection of incompatible donors. Minimizes risk of hemolytic transfusion reactions. Disadvantages. Requires well-maintained reagents. May give false results in cases of autoagglutination or weak antigen expression. Section 3, Antisera, Preparation and Standardization. 3.1 Preparation of antisera. Antisera are solutions containing antibodies used to detect blood group antigens. Source can be derived from human plasma, monoclonal or polyclonal, or produced in animals. Purification removes unwanted proteins. Preservation, additives such as sodium azide prevent bacterial growth. 3.2 Antiserum titration. Determines antibody concentration in a given volume. Helps assess potency and effectiveness. Performed by serial dilution. 3.3 Avidity, potency, and specificity. Avidity, overall binding strength between antigen and antibody. Potency, effectiveness of the antibody in producing visible agglutination. Specificity, ability of antisera to react only with the intended antigen. Section 4. Plant lectins in blood grouping. 4.1 What are lectins? Lectins are plant proteins that can agglutinate specific red cells by binding to certain carbohydrate antigens. 4.2 Dolichos biflorus. A plant whose seeds contain a lectin specific for the A1 antigen. Useful in differentiating A1 from A2 subgroups. 4.3 Preparation and standardization of plant lectin antisera. Extracted from seeds by soaking, grinding, and filtration. Standardized against known red cells. Stored under refrigeration. Section 5. Anticoagulants in blood collection. 5.1 Common anticoagulants. ACD, acid citrate dextrose. CPD, citrate phosphate dextrose. CPDA1, CPD with adenine to extend shelf life. 5.2 Mode of action. Citrate, binds calcium, preventing clotting. Dextrose, preserves red cell metabolism. Adenine, maintains ATP production for red cell viability. 5.3 Side effects. Citrate toxicity in donors if large volumes are transfused rapidly. Requires careful mixing during collection to avoid microclots. Section 6. Blood collection containers. 6.1 Blood bottles, MRC. Glass. Reusable after sterilization. Heavy and fragile. 6.2 Plastic blood bags. Single, double, triple, or quadruple for component preparation. Lightweight, disposable, safer. 6.3 Advantages and disadvantages. Bottles, reusable but fragile. Bags, safe, easy, but single-use. Section 7. Donor screening methods. 7.1 Copper sulfate method, CuSO4. Measure specific gravity of blood. A drop of blood sinks in copper sulfate if hemoglobin is adequate. 7.2 Other screening methods. HemoQ analyzer. Cyanmethemoglobin method. Automated hematology analyzers. In this module, we have built a strong foundation in the science of blood groups, antisera, anticoagulants, and donor screening. These are essential skills for safe and efficient blood transfusion practice.